Okay, so for decades, anyone thinking about how brain cells talk, how that neurotransmitter actually gets out? Well, it was always this debate, right? Either a quick kiss and run or a kind of wasteful full collapse of the vesicle.
Yeah, those were the two main
But
camps.
this new paper, wow, it basically says it's neither, or maybe it's both, but different. They found this unified pathway in hippocampal synapses they're calling kiss shrink run.
And the really counterintuitive bit is the shrink part, the idea that the vesicle actually gets smaller, like halves its size, while the fusion pore is open, releasing the transmitter.
That's wild. So what was the big hurdle? Why did it take so long to figure this out?
Well, you're trying to watch something happen in incredibly fast milliseconds and incredibly small nanometers. You need to see the structures as they change in a working synapse. That's technically very demanding.
Right. You can't just take a regular microscope picture.
Exactly. You need that nanometer resolution in space and millisecond resolution in time. So they had to basically invent a new way to see it.
And that's where this fancy new technique comes in, the optogenetics plus cryo-ET.
Precisely. Optogenetics lets you trigger the neuron to fire with a pulse of light, gives you that millisecond timing. Then, almost instantly, you freeze it super fast, preserving everything. That's the cryo part.
Flash freeze.
Flash freeze, yeah. And then cryo-electron tomography, the ET part, gives you these incredibly detailed 3D images of the frozen synapse, like 3D snapshots.
So they took like a whole movie's worth of snapshots.
Over 1,000,
Good.
covering the first 300 milliseconds after the neuron fired. So you get this structural sequence of events. It's not just inference anymore. You can see the states.
Okay, so turn on the projector. What's the first thing these 3D snapshots showed? What did the vesicles near the active zone look like?
Well, first, a bit of a surprise. Two main sizes of vesicles popped out, a larger type, around 41 nanometers across, and a distinctly smaller one, about 29 nanometers.
Huh, two sizes.
Okay. Yeah. And they could also clearly see different shapes or states based on how the vesicle was interacting with the membrane. Things like tethered, semi-fused, and the really key ones, pore opened and atta-shaped. These look like the intermediates on the way to release.
Gotcha. So you have these structures, these shapes, but how do you prove they're actually part of the real release process and not just random configurations?
Right. Good question. Functional proof. So first, they use the optogenetics to trigger an action potential, make the synapse fire.
Yeah.
And you saw a clear increase in the number of those semi-fused and pore-open vesicles. They popped up right after the stimulus.
Okay, that makes sense. Triggered by the firing.
But the killer experiment was using tetanus toxin, TENT. It specifically chews up the snare proteins.
The machinery that actually pulls the membranes together for fusion.
Exactly, those. And when they added TENT, formation of those semi-fused and pore-open states just stopped, completely blocked.
Ah, okay. So that really pins it down. These states are snare-dependent, are part of the actual fusion pathway.
No doubt about it.
So let's walk through it. Where does the kiss shrink part happen in this sequence?
Okay, it kicks off with the large 41 nanometer vesicles. They first kiss the membrane, that's the semi-fused state, and that peaks really fast, about 4 milliseconds after the action potential.
4 milliseconds. That's quick.
Very quick. Then, by about 8 milliseconds, they transition. They form that pore-open state, and the pore itself, the opening, it's tiny, just 4 nanometers wide, lipid to lipid.
4 nanometers. That's incredibly narrow, barely space for anything.
It is. And crucially, while that tiny 4 nanometer pore is open and the neurotransmitter is presumably rushing out, that's when the shrink happens.
Vesicle shrinks then.
Yes. The large 41 nanometer vesicle membrane rapidly contracts, shrinks down to become that 29 nanometer small pore open state. It loses half its surface area right there. That's the shrink.
Wow. Okay, so it's kissed, the pore is open, it's shrunk while releasing. Now, here's the question that bugged everyone for years, right? What happens to that little shrunken vesicle? Does it just fully merge, flatten into the membrane like the old full collapse model? Or does it detach, like kiss and run?
Slight pause. Well, the data here is pretty definitive. The vast majority, over 80%, follow the run pathway.
They run.
They run. The shrunken 29 nanometer vesicle closes its little pore, detaches from the main membrane, becoming a small semi-fused vesicle again, and then it moves away from that active release site. That whole detachment and clearing out happens by about 70 milliseconds.
70 milliseconds. So the site is clear again very quickly.
Super quickly. Only a small fraction, the minority, actually go on to fully flatten out into that classic E shape, the full collapse.
Okay, so kiss shrink run really seems to be the dominant way here. What are the big takeaways from this then, beyond just solving the mechanism?
Well, the technology itself is a massive innovation. This whole combo, optogenetics, timed perfectly with cryo-ET. It's like a new rule book for watching membrane dynamics in situ, in their natural environment. It's generalizable.
We could use it for other fast cellular processes, presumably?
Absolutely, and the implication for neuroscience is pretty huge. This kiss-shrink-run pathway looks like the main game in these hippocampal synapses. It neatly reconciles those old conflicting models.
And it explains that speed.
Definitely.
Oh,
It allows for what they call hyperfast recycling. Getting that release site ready to go again in under 70 milliseconds is crucial for keeping up with high-frequency signals. And maybe it's also a quality control thing, shunting these smaller, maybe partially used vesicles away quickly.
right. Keeps the main pool fresh. Okay, let's just quickly recap the absolute key points from this deep dive.
Sounds good.
1. Synaptic release, at least in these key memory-related synapses, mainly uses this kiss-shrink-run mechanism.
2. The fusion involves the vesicle rapidly shrinking 41 nanometers down to 29 while it's releasing transmitters through a tiny, tiny 4-nanometer pore.
And 3. This whole pathway explains how synapses can be so efficient and recycle incredibly fast, ready for the next signal.
Exactly. Though it's worth remembering one limitation. This was done in cultured rat neurons. We still need to confirm how general this is across different brain areas or, in a fully intact living brain.
Fair point. Always more to explore. But it does leave you thinking, if that shrink is maybe driven by membrane tension and stabilized by proteins around that tiny 4-nanometer pore, what if you could tweak those proteins?
Oh, interesting thought.
Could you change how reliably the synapse fires, maybe? Its fidelity? Definitely something to ponder.
Yeah, opens up new questions for sure.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has 3 main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the 3rd goal. Let's go! Let's make great science together.